Good morning, everyone. Welcome and thank you for joining us at today's virtual seminar. Today's topic is Finding the Solution to Disease at Garvan, the second of our two-part series. My name is Michelle Levitt. I'm the Philanthropy Manager at the Garvan Research Foundation, which is the fundraising and marketing arm of the Garvan Institute of Medical Research. I have the pleasure of being your MC for this morning. Today we have Priyanka Kalkani and Joseph Hewitt helping moderate and support the panel. And we're also in for a real treat this morning. We've got three bright superstars on our panel, and we're also joined by our Executive Director, Professor Chris Goodnow. Before we begin, I would like to pay my respects to the Gadigal and Gunditjmara peoples, the traditional owners and custodians of the land on which Garvan and our facilities are located. We recognise their continuing connection to land, waters and culture, and pay our respects to elders past, present and emerging. Just some quick housekeeping tools. If you are new to the virtual world, we will have a Q&A component of this session, and if you just look at the bottom of your screen, there's a Q&A button, and please feel free to type any questions you have, and hopefully we'll have time at the end to get through all of them. We have a lot of new faces joining us today virtually. Welcome. It's great to virtually see you all. And we've also been joined by a lot of our long-term supporters, our donors, our partners for the future, our board directors and our volunteers. Thank you for joining us today. To all our newcomers, we hope you enjoy our your first Garvan seminar. I would also like to just give a quick shout out to all our Garvan staff. Most of you may be aware that we have close to 750 staff here at Garvan, and I would just like to acknowledge them today and thank them for their hard work and their dedication during what can certainly be described as a bit of a challenging 2020. Now it's my great pleasure to introduce Professor Chris Goodnow, Executive Director of the Garvan Institute of Medical Research. Chris is a world-renowned immunologist who joined Garvan in 2015 as Deputy Director before becoming Executive Director in 2018. Chris is dedicated to understanding the cause of immune disorders and developing more effective and personalised treatments. Scientifically, Chris is best known for integrating molecular genetics and genomics with immunology. Chris is also currently leading our HOPE research program, which is a transformative research program looking at over 40 autoimmune diseases, which is part of our Garvan-Weizmann partnership and very generously supported by the Bill and Patricia Ritchie Foundation. Today, Chris is going to introduce us to some of the ways that Garvan scientists are discovering the solutions to disease. So please join me in welcoming Professor Chris Goodnow.
Thanks, Michelle. Thanks very much. And look, everyone, welcome. I wish we were all together in the auditorium the way we like to run these seminars, but I think it's the absolute right thing that we're doing this by Zoom. We're sharing information. We're not sharing viral RNA. And look, I hope you enjoyed the seminar, the previous seminar that we had a couple of weeks ago and the ones before that. I think you'll really enjoy the lineup today. Again, you're going to hear from people you haven't heard from before, who will be talking from some of the things that are going on in our Garvan's research centres. So the Garvan has some of the most sophisticated research centres in the world, and the whole purpose of these centres is to connect different research disciplines, to break down some of those traditional silos, and also to work to create bridges and collaborations and partnerships with other research institutes around Sydney, around the country, around the world, with our clinicians, both here at St. Vincent's and in hospitals across Australia, and with the community. And I think you'll get bits of that in the very short talks that you'll hear today. Now, a key thing about each centre is cutting edge technology. And there is this beautiful relationship between scientific discovery, health advances, and technology, cutting edge technology. Yeah. Really, as you all know, technology drives progress, provided it's used by maestros that know how to play those instruments. And that's really what our researchers and research centres are about. So in essence, each of our research centres has been established to support the Institute's overall mission, which is to harness all the information encoded in our genome so that we all live a longer, healthier life. You're going to hear about 3 today: the Garvan Weizmann Centre for Cellular Genomics, the Kinghorn Centre for Clinical Genomics, and the Centre for Targeted Therapy. And I'll introduce a little bit more about each of them before you hear from our star speakers. Now, before we go on to the specific centres, one little piece of news that I wanted to give you a heads up on, and that is that we are also establishing a new centre called the Centre for Population Genomics. It's being led by Daniel MacArthur. Daniel previously served as the co-director of the Medical and Population Genetics program at the Broad Institute of MIT and Harvard. He is arguably the world's most accomplished human large-scale population genomicist that there is. And so we're really thrilled that he's joined Garvan and the Murdoch Children's Research Institute to start setting up this centre as a joint project between Garvan here and the Murdoch Children's Research Institute in Melbourne. There'll be much more to tell you about as that centre starts to be developed and we move closer to opening it. But let's talk about the wonderful centres that we've got running at the moment and the people we're going to hear about. Michelle, did you want to jump in here for a second, or should I press on, do you think?
No, I'd love to. Just before I get you to introduce the Garvan Weizmann Centre for Cellular Genomics, I did have a quick question I thought we'd start off with. Would you be able to explain why genomics is an important area of research to focus on?
Yeah, thanks, Michelle. That is, I mean, you know, people sometimes say, what's genomics? Is that the study of garden gnomes? It's not. It's the study of all the genes and the molecules encoded in the genes that make every cell in our body, which in turn makes every tissue. So, you know, if you think about it, every cell, every body, in fact, one of my former PhD students did some calculations on how many different combinations of molecules are encoded in our genome. It's an absolutely astronomical number. And the challenge for our body is to use the right combinations of molecules to make a brain cell or a blood cell, etc. And errors in those molecules or combining them in the wrong way can change the function of cells. And that's really at the root cause of many of the major unsolved diseases. Cancer, of course, immune and inflammatory diseases, diseases of aging, so in the bones, such as osteoporosis, in the brain, such as Parkinson's disease, and in our metabolism, such as diabetes. So the crucial thing is, at the moment, we have fantastically powerful technology to be able to sequence the whole genome of a person, and that we'll hear a little bit more about how powerful that is from Lisa Ewans in a moment. But it's a long way between having the genome that we inherit and whether we're healthy or diseased, and whether or not we're going to respond to a targeted drug or not. In between that are those single cells, which at the end of the day are the fundamental decision or switch point. And it's only in the last few years that the technology has been available to actually look at how the genes are switched on and what combinations of molecules are actually being produced in individual single cells in any given part of our body. And that's what's called single cell genomics. And the Garvan Weizmann Center for Cellular Genomics is at the absolute cutting edge of that. In fact, it's sequenced the third most single cell genomes of any center in the world, right behind the Broad Institute in Boston and the Sanger Institute in the UK. So we're very thrilled that at the end of last year, we were able to recruit Dr. Sara Ballouz from the Cold Spring Heart Laboratory in New York to take up a role as a group leader in Garvan's single cell and computational genomics lab. And Sara is really focused very much on the extraordinary amount of data that comes from analysing not just genomes, but the way the genome is switched on in thousands, millions of single cells. But she's interested in understanding the genetic architecture of disease, why immune disorders are more frequent in women than men, with the hope of developing targeted treatments in the future. So Sara, I'll hand over to you if I may.
Great, thank you. Hi. All right, thank you for that lovely introduction and summary, Chris. And it's a pleasure to speak to you today. I'm going to talk to you about autoimmune disease and this curious phenomenon on why women are more affected and how we think it's related to the X chromosome. So autoimmune disorders are a range of diseases caused by the immune system attacking the body. The immune system is our line of defense against bacteria and viruses, but when it goes wrong, it starts attacking healthy cells, thinking they're the foreign invaders. And approximately 12% of the population is affected by an autoimmune condition. And so something else that's interesting is that autoimmune diseases are highly sex-specific, so with women being more susceptible and up to 80% affected female. For example, in one of the most imbalanced of these, Sjogren's syndrome, which is characterized by dry eyes and a dry mouth, for every male affected, there are 16 females. And so why is this the case, and why is this the case in other disorders? Well, men and women are different, and the biggest difference are the sex chromosomes. So as Chris sort of mentioned before, chromosomes are the pieces of DNA that hold our genes, and we have 23 pairs of them in every cell. One pair is special though, and these are the ones that determine sex. So males have an X and a Y, and females have two X. And the X chromosome also holds many immune-related genes. We also kind of know that men and women have slightly different immune systems. And so one hypothesis that we have is that there may be something in the way that the X is regulated or like switched on and off at the individual cell level that might be causing this. Now, originally, we couldn't really study this because we were averaging away a lot of this information when we were doing our sequencing by looking at many cells at once. But now with single cell genomics, we can look at individual cells. So this is similar to say that's taking some fruit, putting it into a blender, and making a smoothie. We can guess what's inside. We can sort of tell which piece, we can tell if the piece of fruit went bad, but we can't tell which piece of fruit went bad. But now with single cell, it gives us a fruit salad, and we can sort of easily tell apart the fruit that we see. So although we're not studying fruit, we're studying people and immune cells. Here at the Garvan Weizmann, we use this technology to survey the immune system of an individual. And also Chris mentioned this before about this amazing cohort that we have where we've sequenced 1,000 individuals, both male and female, and thousands of their cells. And this is the 1K cohort. And at the time, it's around 1.2 million cells. And this is led by amazing efforts here by Joseph Powell and his very, very talented team. Which has allowed us to survey these immune systems so we can look at healthy individuals, which will help us explore disease patterns. So as a very quick summary, we use single cell genomics to study the immune systems of both men and women, and then we will focus on the X chromosome, hopefully by exploring this data and lots of data at this level. We can study the population, individual, and cellular level, and that will help us understand not only autoimmunity, but the effects of the X chromosome, discover risk factors, and hopefully be able to diagnose and work towards sort of personalized treatments, basically using cells as a diagnostic tool. So with that, I'd like to end, and I'd also like to take this opportunity to thank all of the team members and support at the Garvan who make this work possible, and Bob and Ruth McGee for their generous donation that funds my position. Thank you.
Thank you, Sarah. That was great. Chris, I'll now hand over back to you to introduce the Kinghorn Centre for Clinical Genomics.
All right, and just to remind everyone that we'll have plenty of time for questions after we hear our three talks from our three speakers, and you can post those questions using the Q&A function at the bottom of the Zoom screen, and we'll be keen to get all your questions. So let me introduce, before we hear from Lisa Ewans, the Kinghorn Centre for Clinical Genomics. It's focused on accelerating and translating genomic discoveries to benefit patients. And its research and its implementation groups focus on translation of genome informatics, genome technologies, and genome education. It involves state-of-the-art equipment, award-winning computer pipelines for genomic analysis, and some really world-leading experts that bridge the world of bioinformatics with the world of clinical pathology, and that's really quite unique. You know, that probably comes together most clearly with Dr. Lisa Ewans. So Lisa is a clinical geneticist at Royal Prince Alfred Hospital and at Sydney Children's Hospital Randwick. So Lisa is really on the front line of what genomics can do or needs to do so that we can all live a longer, healthier life. Lisa is also completing her PhD here in the Garvan's Kinghorn Centre for Clinical Genomics. And Lisa's focus is on the diagnosis and management of rare genetic diseases. So Lisa, thank you very much for speaking this morning.
Thanks so much, Chris. And it's such a pleasure to have the opportunity to talk to you all and discuss my research today, which really is looking at the diagnosis of rare genetic disease and translating that to improve patient care. But before I talk about that, to help you understand the importance of this, I just thought it might be good to take a moment to imagine that you yourself or a close family member has a serious disease or a group of diseases that none of the doctors can put a name to. And if you have a name, they can't find what the genetic cause is. And so you're left not knowing exactly whether it's the right diagnosis or whether this could happen again in a family member. And this is something that many people with rare genetic diseases face. So these conditions occur in less than 1 in 2,000 individuals. And in some rare diseases, this may only actually be in a handful of people around the whole world. In total, there are almost 6,000 rare genetic diseases affecting about 5% of the population, which is over 1 million Australians. And the importance of rare disease has been recognised this year through publication of the government of a National Strategic Action Plan, which is fabulous. And really diagnosis is the key first step. But because there are so many of these diseases, it can be quite difficult. How people present to their doctor in terms of the different kinds of features can be quite hard to make a diagnosis. And for those individuals, we really have a problem in diagnosis, which has been really transformed in the last 10 years with the evolution of what we call genomics, which has been talked about already this morning. And the scale of the problem is really big in rare genetic disease because we have 3 billion letters of genomic code. All of us carry this in each cell in our body. And what we're trying to identify to make the genetic diagnosis is 1 or 2 changes. So those errors that Chris was talking about in that 3 billion letter code. So you can see it's a really big data problem. And it's a little bit like looking for a needle in a haystack. And at KCCG, I've been applying whole exome sequencing and whole genome sequencing to individual samples, their DNA, for people who are thought to have a genetic disease but don't have a definite diagnosis. And my role is in the variant interpretation step to try to identify that single cause for their disease. And we've been really successful. Over half of the people that have been referred to us, we've got a diagnosis and we've been able to translate that back to the patient and to the clinician. And for those people, many of those would not have had a diagnosis without this technology. So let's just have a think about what impact that diagnosis has. Well, initially it resolves that uncertainty, what the condition is. So it gives a bit of information about what might be expected for the future health of that individual. It enables them to connect with other people with the same diagnosis and share experiences and knowledge. And those people might be overseas, for example. We can avoid other investigations, and we can also give more accurate information on whether this could happen again in a family member. It improves our ability to manage the patient and also that can sometimes, for a small proportion of people, lead to a new therapy, a new treatment. So just in summary, diagnosis is really important for millions of people who live with rare genetic disease around the world. Genomic technology has revolutionized diagnosis, and that's really essential to translate to improve patient care. And as others have said, lots of thanks to lots of people. I can't name them all, but particularly the patients, families, clinicians, the KCCG and UNSW. Thank you.
Thank you, Lisa. That was fantastic. I'll now hand over back to Chris to introduce us to the Centre for Targeted Therapy. Thanks, Chris.
Thanks, Michelle, and thanks, Lisa. You know, I wish we had more time. Lisa, I know Lisa has some extraordinary stories as case studies, and maybe at question time we can tease those out. Look, let's talk now about the Centre for Targeted Therapy, which was established as a centre of excellence to advance the development of a new class of biological drugs, which are human monoclonal antibodies. And Peter Schofield is a real expert at this, and he will talk about those in more detail, but the focus is on improving the transition of basic research, immunology research about antibodies, and actually turning that and even bypassing the variability in our normal immune system to produce antibodies as drug candidates, products, and as treatments for disease. So, Peter, thank you very much for speaking this morning too.
Thanks, Chris, and thanks everyone for tuning in. So I'm just going to launch straight into what our lab's area of expertise is, and that is antibodies. And I'm sure everyone has a vague idea already about what an antibody is, and probably have heard about them a bit more in recent times. But basically, an antibody is like this protein you can see here on the screen, a T or Y-shaped protein. And all of us have billions of different antibodies floating around in our bloodstream right now. And they're giving us protection to things we've either been immunized with or have previously been infected with. And that's one of the key elements of our immune system. And how you should really think about what this T-shaped protein is it's like having a pair of hands here. And these hands are able to bind very strongly and very specifically to incoming pathogens to the body. And once they grab a hold of this incoming pathogen, this other portion of the antibody acts like an emergency beacon, which will recruit the rest of the immune system to come and wipe that guy out. So you can see Mr. T cell has turned up here. And this is a really crucial element of the immune system. But innovations over the last 30 years or so have allowed us to harness these amazing little proteins and engineer them in the lab to become precision medicines. And surprisingly, a lot of these medicines are actually being used as cancer therapies and therapies for treating autoimmune diseases or inflammatory diseases such as arthritis. And it's really important that the Garvan has this area of research as one of its key focuses because they've really exploded in the last 10 years. So what I've got here is a table of the top 20 drugs by sales in 2016. If you looked at this in 2006, there wasn't a single antibody there. But nowadays, we've got at least 6 of the top 20 drugs are antibodies. And all these antibodies here are actually being used to treat different inflammatory diseases or cancers. And that's usually what we're targeting as well. And we are still targeting those classic targets. But this year, for obvious reasons, we've also pivoted our research towards a very important target right now, which is COVID-19. And so basically what we do in our lab to try and come up with a therapy for this virus is we take our libraries of antibodies we've developed, and these have about 1 billion different antibodies in them. All of them have different sets of hands with different specificities of what they can bind to. And what we then do is we pan them against a target. It's like panning for gold. We're looking for the antibodies that can bind to a target. In this case, what we've got is a protein which is right on the end of the spike of those viruses. So once we isolate and identify the antibodies that can bind to this spike protein, we then are collaborating with other research institutes to to test whether this this antibody can actually kill the virus in a very real way. Ultimately, the end goal for the desired outcomes for this kind of thing, everyone knows that we're trying to develop vaccines against coronavirus, but that is proving quite tricky. This is another mode of therapy that could be done, and instead of immunizing someone and making them make their own antibodies against COVID-19, this antibody could be used to treat patients that actually have a very critical condition from COVID-19, and it'll basically bypass the need for them to develop their own antibodies to it, and should be able to kill the virus in these people. And it could also be made at this industrial scale, not just to treat these kind of people, but could also be given to frontline workers as a protective thing. But it wouldn't have long-lasting immunity in these people. They would just have an antibody in their system for a certain amount of time that would protect them from coronavirus. So those are the two ways that these antibodies could be used, and that's what we're really working around the clock on right now. So.
Fantastic. Thanks, Peter. All right, I think we're up to our Q&A component of the session. So I'll ask the panel to please join me back on the screen. We have some great questions that are coming through. Just going to grab the first one, and I think I might pass this one on to you, Chris. Can genomic analysis at Kinghorn be requested by individuals, or is it only medical professionals?
Yeah, thanks. Thanks for that question. It's a really good question, and the short answer is no. Garvan's a research institute, and so we focus on research. Testing itself is really, once research has actually created a test, we like to pass that baton to a pathology service, for example, or to one of the hospitals. So at the moment, genomic analysis at Kinghorn is part of large clinical trials or clinical cohort studies. But it's more than that. And Lisa, do you want to maybe talk about examples where, maybe one example where that sort of brings some life into how you, as a clinical geneticist, have used the Kinghorn Centres for Clinical Genomic Analysis to help inform you on how to manage a case?
Yeah, sure. Well, we've got, I think, ones that have therapeutic changes is rare. That's probably more immunological disorders, actually, where you can find an immune deficiency, for example. But in our cohort that we've looked at, we have been able to make new diagnoses or alter diagnoses, which changes the health surveillance outcome. So one particular patient was thought to have a metabolic condition and had some treatment for that, but it turned out to be a genetic condition which she didn't need the treatment for, so that could be looked at stopping, and there were health issues that we could look for. And then another family I can think of off the top of my head had two boys affected with a neurological condition, and they had family members who were women, and people were worried that this was an X-linked condition and could be carried by a woman but not have the disease and passed on. And our genome sequencing identified that they actually had a recessive disease, which meant that the women wouldn't be passing on the condition. And actually it had a metabolic component, and one of the boys became very sick and ended up in pediatric intensive care. And because we knew the genetic diagnosis, we didn't have to try to understand why this was happening. We knew that this could happen in this condition, and that boy could have his treatment focused on in that setting. So that's the kind of thing that we've been able to do with our diagnosis, which is just really great.
That's really beautiful, Lisa, because it really brings out the fact that doing the genomic analysis is a bit like drinking from a fire hose. As you said, 3 billion letters. A lot of them vary from one of us to the next, and the Kinghorn Centre for Clinical Genomics can whittle that down through all of their computational pipelines to a very short list. But then there needs to be a very important partnership with the clinical geneticist, yourself, and the treating doctors to interpret that information in the context of everything else you know about the family and the patient.
Yes, exactly. And I think particularly for the patients who present with a complex presentation. So they have different organ systems involved. And it's clearly, we think, genetic because a genetics health professional has assessed the individual. But because there are so many diseases and so many diseases being newly described, it can be really difficult on a clinical basis alone to make the diagnosis, which is why this technology has just changed the game completely, actually. And like you say, we partner with lots of different people with expertise, particularly the bioinformatics and the sequencing laboratory, that help us get down to a smaller list of changes to be able to work out and compare the patient's presenting clinical features and compare with their genetic features and try and marry the two together.
Thanks, Lisa.
Thanks, Lisa. Just another follow-up question on that. A question has come through. How is a rare disease classified as genetic when we don't know where or why the illness happens? Example, neurological disorders.
Lisa, if you would
take that one.
Yes,
yeah, Lisa.
Sure. So I sort of touched on that a little bit before, in that in the way that the health system currently works is that the people trained to answer that question are really working in clinical genetics. So there's people like myself, clinical geneticists, and we work closely with genetic counsellors, and we receive referrals usually from doctors who are considering a genetic condition. Sometimes the patient looks it up and thinks they have it, and then they get a referral. And we, I guess, have a framework in the way we assess people. Some conditions are known to be genetic. Some people present in a way which we're trained to go, look, this looks to be genetic. There are some really rare diseases which we know are not genetic and are caused by different things, but it can be a bit difficult to differentiate. But I suppose you have trained health professionals to look at that question.
Yeah, so if I might jump in there, one of the things that certainly in my own field of immunology that we've seen happen at an ever increasing rate in the last 10 or 15 years is diseases that were believed not to be genetic and not to be a rare genetic defect. For example, a rare viral infection of the brain turn out to be rare genetic defects once you have the technology to look at people's genomes and interpret what that means. Doesn't mean all diseases are, but Lisa, could I ask you just to go one step further? You mentioned that in some cases, not only do you, provide a diagnosis and connect people with around the world with other cases and expert clinicians, but sometimes you actually are able to, through the diagnosis, connect a person with a rare disease with a treatment that really almost cures them. Or does effectively cure them. Can you share an example of that?
Sure. So I haven't personally been involved in any cases like that, but I know through the Kinghorn Centre for Clinical Genomics that has happened for a young boy who had an immune deficiency that was identified through whole exome sequencing, and that happened very quickly. And there are stories like this around the world where the immunological genetic defect is identified and a specific targeted therapy can be provided. And this child was very sick and was in the sort of intensive care setting and had treatment, and then now is leading a pretty healthy life. And it has inspired his own family to get involved in this type of work as well.
Yes, thanks, Lisa. That's right. It is, those are the light on the hill cases, and that little boy actually comes into Garvan every so often because, boy, is he a different, a person from what he was when he was in intensive care, and things were looking very grim. Now he's just he's binging around like a rapidly growing boy, and it's just a beautiful example of how that teamwork between the clinicians and the genomic analysis and all the basic science that runs between that. He has to keep getting that injection of that targeted drug that replaces what he's missing every fortnight, but it's given his life back essentially normally.
It's incredible.
Okay, look, thank you, Lisa.
Thanks, Lisa. This is a question for Sara. I wonder if you were looking for single-cell transcriptome only or examining the epigenome as well.
Currently, it's just the transcriptome, but we will be looking at the epigenome as well. So the transcriptome is looking at the RNA that comes from the DNA, and the epigenome is all of the little bits and pieces on top of the DNA. But single cell genomics allows us to do both, and we can use the transcriptome to actually know what the epigenome is as well. So yes, is the short answer.
Okay. Great, thanks, Sarah. I've got one for Peter. How is the antibody made / made of?
Okay, well, that depends in what context, but in our bodies, what's happening is we have a huge repertoire of B cells. And basically, when there is a vaccine or we have an infection, basically, if each B cell is inherently making its own type of antibody, and if it gets a signal, if its antibody is able to bind to this thing that it needs to target, and when it gets those signals, it will expand, and so basically, you can say that these B cells that are getting expanded because they have a good antibody to something are making the antibody that's in our bloodstream naturally. In the lab, we have a range of different ways that we find an antibody that we think is good. But ultimately, at the end of the day, when we want to make antibodies as a therapy for someone, we are still using cells. So we will have a large vat of cell culture that's making this antibody, and then it is purified to be used as a therapy. So it's a protein every time. In the body, it's made by our own B cells. And in the lab, at the end of the day, it's also made in cells. It never becomes fully synthetic. It's just wouldn't be a good way to make it. So,
and I suppose if I could just add, there's the genetic poker machine that lies behind those hands that Peter you brought to life so well is one of the most miraculous things that they're imagining.
All right, we have some more questions coming through. This is another one for you, Peter, and perhaps Chris, you might also like to jump in. Do you know how long an antibody therapy provide immunity compared to a vaccine?
Yeah, we talk. So antibodies, they're excellent at staying in the bloodstream. They've basically there's a whole bunch of things about them that make them stay in the bloodstream longer than a normal protein would, but it's a transient thing, and the focus really would be is to think of this as a therapy for someone who's already sick with coronavirus, rather than something that you would take now to keep yourself safe for a number of years. But yeah, whereas when you get vaccinated, if it's a successful vaccination, you will have B cells in your system that will remember that target and know that their antibody is the good one. So if it ever comes along as a real infection, they'll start expanding straight away and making the antibody you need. So it's a more transient thing than a vaccine, but it could be extremely effective at wiping out the virus if it's needed as a therapy. So.
Chris, do you have anything to add on that?
Peter's answered it very well. About a month is the very short answer after a single injection of an antibody. As Peter said, if your own B cells are making it, they'll keep making the antibody. The legendary example from the Faroe Islands with measles is, which is one of the most successful vaccines. Your B cells will keep making the antibody for 20, 30, 50 years, but they'll be constantly replacing the antibodies that are turning over. Any preformed antibody in your bloodstream on average about 3 to 4 weeks. So for example, there's an antibody which is given to premature babies and babies at risk to prevent them from being infected with a respiratory virus called respiratory syncytial virus, and that's a monoclonal antibody, and it's usually given as a single injection and provides about a month's protection for the premature babies. Have I got that right, Peter?
Yeah.
Great. Chris, this might be a one for you. I just Lost her. There it is. This is relating to the autoimmune disease work, and Sarah, feel free to also jump in, but are there any advancements on diagnosing chronic fatigue syndrome and or any treatments?
Yes, so that is a really important question. It's one that we spent quite a bit of time as part of the HOPE research project initiating on the hypothesis that it might be an autoimmune disease, chronic fatigue syndrome. It is a really terrible disease and a real mystery, medical mystery. We got very excited because there was preliminary clinical trial evidence that a monoclonal antibody that treats autoimmune disease called rituximab, one of those targeted antibodies that Peter had on his list of top 10 drugs, was being effective for chronic fatigue syndrome. But when the full proper randomized controlled trial results came out about a year ago, it turned out it had no benefit at all. So, this is the problem in research. You get your hopes up, and then the data is the great leveler. So at this stage, we're watching that space very closely, but my own reading of literature is it's still every bit as much a mystery as it was before. It's sort of the Mount Everest of diseases at the moment.
Thanks, Chris. Lisa, I'll throw this over to you. How much international collaboration is taking place in genomic analysis? The vastness of the field must make it essential to share knowledge.
Absolutely. That's one of the strong focuses in the genetic community over the last few years. And we have programs that try to look at individuals for genetic diagnoses that have gone through exome sequencing and or genome sequencing and still don't have an answer. And that really comes down to collaboration. One of the families we diagnosed had what we call an ultra rare disease, so less than 1 in 2 million people around the world with it. And we actually took the opportunity to curate all the known data, do some collaborations with scientists and laboratories around the world, and get a paper together to put that out to the medical community. And we have a tool called Matchmaker Exchange, which draws in a lot of different tools, including a data repository to put genetic and genomic variants into, which people can then link patients around the world and compare clinical features. We have ways for patients to connect with each other. There are ways for, so both the feature sharing and the genetic variant sharing. And many of us in the clinical practice now, when we have a patient who remains undiagnosed, we put some of their data with, of course, consent and anonymized into these systems, and then we can get to diagnoses that way. And I've got many colleagues who have identified new rare disease genes in that way.
Great, thanks, Lisa. Chris, this one is for you. In your opinion, will any of the COVID-19 vaccines in clinical trials be successful?
Oh dear, that's such a tough question. First, I'll answer it as a member of the community. Gosh, I hope so. We sure need one of them to come out a winner. As an immunologist and a scientist, I, for example, poured through the detailed data and supplementary data, everything that was in the 2 papers that came out in the Lancet earlier this week, reporting on the first phase of the human trials of the Oxford vaccine and a similar vaccine from China. My own assessment is that the good news, which was what we heard in the newspapers, was that they did induce some of those kinds of antibodies that Peter talked about that have the hands that actually not just grab the virus, but neutralize it and inactivate its infectivity. The bad news is the level of those antibodies in the blood were worryingly low. They were half the levels that are on average present in someone that's just gotten over COVID-19. And we already know those levels are worryingly low and may not provide much, if any, immunity to reinfection. So the time will tell. It'll be much, much longer before we know whether the levels of antibody that these vaccines are inducing are enough to provide some people to be resistant to infection, to slow the spread in the community. That may be the case. What we know from all the coronaviruses is they've got some tricks up their sleeve that allow them to elicit very low levels of these neutralizing antibodies compared to influenza or measles. Something special about the whole class of coronaviruses, which is one of the things we're working on here at Garvan and to try and crack. It's probably something to do with that spike protein structure. And the other key thing that the coronaviruses seem to have mastered is that the antibodies they do elicit don't persist. Our B cells don't keep making them for very long, so that within a year after having a common cold coronavirus, your antibodies are no longer present in your blood, and you're no longer immune to being reinfected with the exact same cold virus once again. For SARS, no one was ever tested as to whether they could be reinfected, but we certainly, there was really good literature that 2 years out from having had full-blown SARS and having had a lot of antibody that, in a cohort of 54 people that were followed in China in 2003 to 6, that pretty much all of them had lost almost all of their detectable circulating antibody, and so presumably would be susceptible to reinfection. So the good news is we've got an awful lot of shots on goal around the world in the vaccine field. The concerning side of it is that at this stage, they don't look like the kinds of vaccines we would really need to stop the spread of the disease through communities around the world.
Okay, thanks for that, Chris. Peter, the next one's for you. Is it possible that the corona spike protein that the antibody binds to exists elsewhere in the body, presenting an autoimmune situation?
A really good question. Viruses spend a lot of their time trying to look like us. So there's always these elements of them that are trying to resemble things that are present already in the body to try and help them evade an immune response. In terms of breaking it down to the type of antibodies that I was explaining earlier, these will be individual antibodies with one set of hands that are known to bind in a very specific way, and ultimately would be tested to not be reacting with anything in the body. But for sure, there can be some antibodies in some viruses that are elicited, and you may end up with an autoimmune disease. That's a possibility, but I don't think that's the primary concern at the moment. I think virus is a more important concern. So. And Chris can probably add in more excellent answer to that as well, I would say. But.
Yeah, so we and Peter's been part of this project. We've had some support as part of a Gates Foundation project to try and crack the problem of a vaccine for HIV over the last couple of years. There's been a massive worldwide effort on that. All of the attempts at vaccines, as you know, they get to this point that they look a little bit promising for HIV, and then they don't pan out because you just don't induce enough antibody of the right types. And the answer there, what's come out of that body of research, particularly from other groups that we can't claim because we've come in relatively late to that here at Garvan, but is that the HIV virus is a master of cloaking itself, as Peter said, so it has a foreign virus protein, which our immune system and our antibodies should just latch onto with ease. But it cloaks the whole of that virus protein with our own sugars, the sugars that coat all of our normal proteins. And so it makes it very hard for an antibody those hands to grab the virus and not also be constantly grabbing every other protein that's on the surface of all of our cells. And so my own work for the last 25 years has documented all the breaks or checkpoints in the immune system that make sure we don't make those kind of antibodies. And the virus, HIV, has mastered those checkpoints to essentially evade our antibody production. At this stage, it doesn't look like the coronavirus spike is using that strategy. So I think it must be using another strategy, and that will be one of the things that should come out of a lot of the research that we're doing within Garvan and in partnership with people around the world, is to try and figure out what is this strategy. It seems to be an ancient strategy that is shared by most or all the coronaviruses, but at this stage, we don't know what it is.
Thanks, Chris. Sarah, this one's for you. Do you think a time will come where we will be able to test for autoimmune diseases before symptoms show?
I hope so. I think this is key. So we already use sort of like blood as a diagnostic in like many tests, and it just seems like why not? Like if we can sort of see the pre-autoimmune condition before it sort of like progresses, then diagnostically through blood, then yes, I think over time, like that's the sort of end goal of all of this. And hopefully we can find sort of like biomarkers or maybe even different cell types that are around, and that will help us help it become like that diagnostic sort of form. Yeah.
Fantastic. I've got a good question that I thought I could throw to each of the panel members. The question is, hi, I'm a first-year undergraduate student currently doing a research project with the Weizmann Institute. My question is, what has been the most exciting breakthroughs and where do you see your field going in the next 10 years? So I thought that might be a nice one for each of our panel members to answer. Maybe Lisa, you might like to start?
Sure. It's hard because it's such a broad thing that we do. Every time there's a little breakthrough in one component, we could see how that might work for a lot of different people. So for example, the work that Sarah's lab does that looks at the single cell genomics, we hope that that type of work can help us understand what's missing for the people that we don't diagnose, where are those missing diagnoses coming from. And I suppose just in general, the gene therapy approach that's coming out with new technology that's been able to really target the specific error in the genome that causes a genetic disease, and the ability to be able to hopefully provide a therapy that can fix that. And maybe if we apply that really early for people, that we might actually be able to change some of the outcome for those people. I think for us, that's very exciting, but it's so limited at this stage.
Great, thanks Lisa. And what about you, Peter?
I'd probably say CAR T cells are a very exciting thing that's come in the last 10 years. So, and it's really a cancer therapy, and it's kind of similar to an antibody, but what you got to picture now is you're taking T cells from a patient, often someone who's got a blood cancer, and then these T cells, which are part of the immune system, you're sort of almost putting a small antibody on them that primes them to go and target the blood cancer cells. And you can have patients that have, a completely unsolvable disease in the other way, and these CAR T cells can just wipe out that cancer very dramatically. So that's a really exciting one.
Great, thanks. And what about you, Sarah?
I might geek out a little and say, I'm really excited by the technologies and the scale that it's has achieved. So we used to only be able to look at hundreds of cells, and now it's to millions. And a lot of our computational power has also increased. So the breakthrough in the technology is just as exciting as the science itself. So I think being able to sort of merge those two has been really, really great. And just to bring back to an earlier point on international collaborations and just data sharing, and I think that is also really key, and that's made a lot of people able to have these breakthroughs because we're able to share a lot of our data. So yeah, that's my percentage.
Fantastic. And Chris, what about you?
Yeah, for me, it is a tough question. There's been so many things, but I think that the thing for me For example, I joined the Garvan Institute only 5.5 years ago with this idea that all this fantastic basic immunology discoveries that I'd participated in from my career might actually be useful for patients with immunological disorders. But what's happened really just in the last 5 or 10 years is that's exceeded even my wildest expectations. The ability to sequence the genome of people and not only understand why they've got an autoimmune disorder, but actually match it up with a targeted treatment. And we talked about the little boy. There are many other examples like that. That didn't happen. To put a point on that, I used to run the immunology lectures for the medical students at Stanford University when I was on the faculty there. And one of the complaints that the medical students had was that we kept introducing more and more characters onto the immunological stage, and it was horribly complicated, this immunology. But at the end of the day, the treatment for everything was cortisone and poisons. And so what was the point of just, telling them all this stuff when it really wasn't going to be useful for them on the clinic? And that has completely changed in the last 10 years. The checkpoint inhibitors for cancer that are used on a daily basis for patients that are coming into the Kinghorn Cancer Centre, just drawing on all those characters that we used to introduce on the stage, but now they're actually useful. So if I was running the immunology course at Stanford now, those complaints would be gasps of excitement, and that's progress.
Great, thank you so much, Chris. All right, I'm sorry to say that we've run out of time for all the questions. If you would like to send them through, please do so to Garvan, and we'll do our best to get to them at a later time. I would like to give a very warm thanks to our amazing panel today. Thank you so much for sharing your amazing research in such a short amount of time. Thank you also to Priyanka and Joseph, who work behind the scenes to make sure these seminars run smoothly and always do a fantastic job. And most importantly, thank you also to everyone who joined us today, to our incredible supporters and our partners for the future. Thank you so much. There will be a short survey that will pop up on your screen shortly. We would really value your full and frank feedback so we can continue to improve these seminars and make them as informative and valuable for you. Thank you also to all of you who have already made a donation to Garvan this year. We are so thankful for your support, and it is so critical and transformational in the work that we do. If you have been particularly inspired by today's seminar and would like to make a donation, please do follow the link. It will appear on your screen shortly too. We hope you take care and that you and your family remain safe and happy and healthy. And we thank you so much for joining us today, and we hope we'll get the chance for you to join a future seminar shortly. Thank you so much and take care.